working unit, if you will, of the kidneys is a structure called a nephron. And nephrons are extremely common in your kidneys. In fact, you have over 1 million of them in each of your kidneys. And there are 2 different types of nephrons. By far and away the more common type of nephron is something called a cortical nephron. They are called cortical nephrons because they're found up here in the cortex of the kidney, not in a renal pyramid. The other type of nephron that's found in the kidneys are called juxtamedullary nephrons. And juxtamedullary nephrons are much less common, and they are found right at the interface between a renal pyramid and the cortex out here. Juxtamedullary means right next to or adjacent to the medulla of the kidney, okay? The renal medulla is the area where all of the pyramids are located. The cortex, again, remember, is the area surrounding that here, okay? Now, in terms of these different types of nephrons, we are going to learn later that they perform different jobs for the kidney. The cortical nephrons are responsible for filtering waste out of the blood and manufacturing brand new urine. By comparison, the juxtamedullary nephrons, the ones that are located immediately in the interface between the medulla and cortex of the kidneys, are more concerned instead with something called water conservation. They don't directly help your body conserve water, but they set up an environment in the kidneys that allows for water to be conserved. We will talk about that later. Now, for now, let's just talk about what are the generalized pieces and parts of a nephron, regardless of whether it happens to be a cortical nephron or a juxtamedullary nephron. This is an example or a drawing here of a typical nephron. And let me see if I have a bigger picture of it. I don't. Okay. The nephron is going to have a number of parts to it. First of all, it's going to have an incoming blood vessel called an afferent arteriole. That afferent arteriole is going to come into a capillary bed called the glomerular capillaries. And then that blood's going to exit out through an efferent arteriole. Now, when it's in that glomerular capillary bed here, we are going to have fluids and solutes leaking out, and they are going to be collected by this cup-like structure here, which is called the glomerulus, or the glomerular capsule. Okay, glomerular capsule. And then the fluid is going to filter into this long squiggly tube here. And that squiggly tube continues onward here. It goes quite a ways, okay, all the way out to here. This tubule has some discrete parts to it that I'll get to in another slide. For now, you can call it just the renal tubule, or the renal tubule. And the renal tubule eventually dumps its forming urine into something called the collecting duct here. Okay, collecting duct. Or collecting tubule if you want to. Tubule or duct. Now, also important to the proper function of your kidneys is a set or series of blood vessels called the vasa recta. The vasa recta are the blood vessels that directly provide the kidneys with the oxygen and carbon dioxide they need. Kind of like the coronary blood vessels are what feed the heart muscle itself. So the blood that's coming into these nephrons is not blood that's helping to provide nutrients and oxygen to the kidneys. It's blood that's coming in to be filtered by the kidneys. The blood vessels responsible for delivering items to the kidneys so the kidneys can stay alive are the vasa recta. Now, I want to take a while here and look at the nephron that we talked about, all these pieces and parts that make up the functional unit of the kidneys, the nephron, and go through them one by one. And we're going to start here in this region with that glomerular capsule. And old textbooks, you will see the glomerular capsule referred to as Bowman's capsule because a guy by the name of Bowman first described it. And if we look at an up close of the glomerular capsule, here's what we would see. First of all, there is in the glomerular capsule an outer parietal layer that I'm highlighting in green here, made up of simple squamous epithelial cells. Inside of that is a second layer of cells, and this is the visceral layer. And the visceral layer is made up of these, in this picture, these kind of orangish colored cells that are literally wrapping themselves around the capillaries. Okay? So we have the capillaries in here, and then the capillaries are coated or covered with another layer of cells that we call the visceral layer of the glomerular capsule. And the cells that make up that visceral layer, the names of the cells that wrap themselves around the capillaries, are called podocytes, which literally means footed cell. Podo means foot. So literally it's like these cells have little feet that wrap them, or fingers, if you will, that wrap themselves around the capillaries very tightly. Now, in between the outer parietal layer and the inner visceral layer is a space called the capsular space, and this is where the filtered fluid that's being squeezed out of the capillaries is captured. Okay, the fluid, what's called the glomerular filtrate, that fluid that's filtered out, gets captured in this capsular space, and then it gets fed into the renal tubule here. This black picture over here with the green and blue is a picture of the glomerular capsule with the outer parietal layer removed. So you can see the inner visceral layer, and the green and blue are those podocytes that have wrapped themselves around the capillary bed. Now, once my fluid is filtered out, and let me back up here for 1 second and just remind you that blood comes into this capillary bed through the afferent, begins with an A, arterial, and it exits out through the efferent, begins with an E, arterial. I want you to notice that the afferent arterial is larger in diameter than is the efferent arterial. And that's important because in order to force fluid to get filtered out of the blood and get caught in the glomerular space here, I have to have the blood at a sufficiently high blood pressure to do that. And the easy way to do that is allow more blood to go in than filter out through the efferents. This is, again, that whole idea of, like we had with the lymph node, if I have more lanes of traffic feeding in and fewer lanes of traffic exiting out, is going to cause pressure to build up where the blood is coming in. So as more blood's coming in than is exiting out at any given time, so the pressure in here increases, which means more fluid will be filtered out and caught in the renal tubule. Now let's take a look at the renal tubule and see what pieces and parts constitute it. The first region of the renal tubule is very twisted and turned. Another word for having very twisty, turny tubule is being convoluted. And because this series of twisty tubes is located very near to the glomerular capsule, we call this the proximal, meaning near to, convoluted tubule. Convoluted meaning twisted. Okay, you can call it proximal tubule for short, but the full name is proximal convoluted tubule, or PCT. And this is lined with simple cuboidal epithelial cells with microvilli. And what that really means ultimately is if I have microvilli, I'm really good at absorbing things. Okay, and we will talk about why that's important later. The next region of the renal tubule is this big part that dips down like this. This is called the nephron loop. Old textbooks call it the loop of Henle because a guy named Henle discovered it. This is the portion of the nephron where we try to conserve water. In the descending part, the part going down, notice how it's thin through here. It's called the thin segment, and this is where we conserve water. And then on the ascending part up here, notice how it gets thicker. That's called the thick segment. And this is where we absorb different electrolytes like sodium. We reclaim more sodium and potassium, etc. Okay. The next section is another kind of twisty region, but it's located farther away from the glomerular capsule, so we call it the distal convoluted tubule. And this distal convoluted tubule, although they didn't draw it accurate here, the distal convoluted tubule actually has a part of it that nestles back over here close to the Bowman's capsule and then comes back over. And they didn't draw that in there as they should have. But we will learn later that in this region where it nestles up close to the glomerular capsule that there is a region called the juxtaglomerular apparatus, and that's where we do some regulation of how much urine is being formed. From here, the urine that's been forming in this tubule enters into the collecting duct. The collecting duct goes into a papillary duct where we're collecting the foreign urine. Then it goes into a minor calyx, which feeds into a major calyx, which feeds into the renal pelvis, which feeds into the ureter, which feeds into the bladder, which feeds through the urethra and out of the body. Now, if we were to look at a more accurate picture of what a nephron looks like, it would look more like this. The tubules are far longer than have been depicted in this picture, and much more twisty and windy. So just keep that in mind. This is a simplification of what the nephron really looks like. All right, now let's start talking about how exactly it is that I form urine. This is a 4-stage or a 4-part process. It begins with glomerular filtration in that Bowman's capsule or glomerular capsule up here. First thing we got to do is filter the blood so that things come out of the blood. Then in the proximal convoluted tubule here, we're going to reabsorb the stuff that we filtered out that we meant to keep. Okay. Then the next thing we're going to do is something called tubular secretion, which is one last chance to get rid of stuff from the blood we don't want to keep. And finally, we're going to do something called water conservation, where we make sure we're not losing too much water in the forming urine. So let's go through this step by step. We're going to start out with glomerular filtration. This is step 1 of urine formation. Again, remember we have an afferent arteriole coming inward. It's larger than the efferent or exiting arteriole, which means that I get an increase of pressure in the capillaries here that are surrounded by the podocytes. And as a result, I get a pushing out of fluid from the blood. Blood plasma gets pushed or forced out of the blood, and along with the blood plasma that's forced out are all the small solutes like sodium, potassium, hydrogen ions, different types of things like glucose in some instances, for example. So all of these items get filtered out of the blood. Larger things like very large proteins or red blood cells normally cannot cross the capillary barrier, so they will remain in the blood and exit out through the efferent arteriole. Now, again, remember that we have this outer visceral layer, the inner parietal layer. We have a space in between. This is where the glomerular filtrate is collected and captured, and it's then fed into the renal tubule, to the first part of the renal tubule, which is called the proximal convoluted tubule. Now, before we go on to talk about the next part of this process, I want to take a minute here and talk about glomerular filtration and how changes in blood pressure can influence glomerular filtration rates. If I suddenly increase the pressure of blood entering into this glomerular capsule, the end result is that I'm going to actually be because I have a higher pressure, I'll be forcing more plasma out of the blood. If my blood pressure remains high for an extended period of time, a problem can occur, because remember that capillaries are very delicate. They're only a single cell layer thick. Now, in this case, in the nephrons, they actually have those podocytes wrapped around them. That helps to strengthen them a little bit, but still, if I have a high blood pressure for an extended period of time, I can actually start to cause tearing and then scarring occur in those glomerular capillaries. So high blood pressure over an extended period of time that's left untreated can actually damage the nephrons and reduce the kidney's ability to do its job. And in fact, in untreated high blood pressure patients that have extreme high blood pressure untreated for long periods of time, they can actually go into renal failure due to this. Okay, so we've got this first process, is glomerular filtration, where everything but proteins and cells can potentially be filtered out of the blood and be funneled into the proximal convoluted tubule. And at this point, we're going to enter into the next step, which is tubular reabsorption. Before we go there, though, I'm going to talk for just a minute more about these podocytes because they're kind of cool little things. Remember again that my glomerular capillaries are very thin, but that they are surrounded by this intervisceral layer of the glomerular capsule, which is made up of these podocytes. And these glomerular capillaries are fenestrated, which, if you remember from our previous discussions in blood vessels, means they have tiny holes in them to allow for more things to filter out more readily. So everything but cells and basically proteins can come out. So here's my fenestrated capillary. You can see it there with all the holes in it, and then surrounding it is another layer of cells, these podocytes, and you can see that they do indeed have all these finger-like projections, and that one podocyte, if I were to draw its finger-like projections like this, interdigitates or coordinates itself with the neighboring podocyte like this, so that the finger-like projections of these two cells meet up with each other as if you were interdigitating your fingers, and that solutes then have to try to pass through the little slits in between that I'm outlining in black here. So the little finger-like projections I've put in blue and green here are the pedicels. Those are the extensions, and that we have these in black here, the filtration slits through which things pass through. Now, what's interesting about these filtration slits is, for one thing, they're very small, so they prevent things from getting out, like large proteins that may have made it through the holes in the fenestrated capillary. When they get to those filtration slits, the filtration slits are physically so small that many of the proteins can't get out. The other thing about the filtration slits that makes them highly useful is that they have a lot of negatively charged molecules in those filtration slits. And as it turns out, proteins are also negatively charged, and things that are negatively charged will be repelled by other things that are negatively charged. So this, too, helps to prevent proteins from being filtered out in the blood. This picture up here shows you a nice real electron micrograph picture of what those podocytes look like with all those little pedicels or finger-like projections surrounding the capillaries. So once I have done this process of filtration, I'm going to have a bunch of different solutes and goodies that have been filtered out of the blood. So the question is, what kind of stuff gets filtered out of the blood? Well, we know that cells do not get filtered out of the blood, and we know that large proteins and most other proteins do not get filtered out of the blood. But the things that can get filtered out include what you see here and what I've listed here, things like water, potassium and sodium ions, amino acids, glucose molecules, urea, uric acid, creatinine, and different types of water-soluble vitamins like vitamin C and vitamin A. Now, if something happens and I damage my capillaries and suddenly proteins start leaking out into the forming urine, which obviously is not supposed to happen, then I have a condition called proteinuria, which means protein is present in the urine. The most common one that would be present is the most common protein found in the blood, which is albumin. If I even have greater damage to the nephrons, such that the holes in the capillaries are so large that red blood cells are entering into the urine, then I have what's called hematuria, or blood in the urine. Now, your kidneys do an amazing job of filtering your blood all the time. In fact, in a single day, they can filter as many as 180 liters of blood plasma. Now, you only have about 5 liters of blood, so that means the same blood is passing through your kidneys, obviously, dozens of times per day. Out of that 180 liters of blood plasma that's filtered per day, we only make on average about 2 liters of actual urine. So most of the fluid that gets filtered out gets actually reabsorbed, and most of the solutes that get filtered out actually get reabsorbed, so that we only end up with about 2 liters of actual urine that is made. So I want to talk now about this process of reabsorption, because it's obviously really critical. We are reabsorbing 178 liters of water and other solutes every day. So let's look at what this involves. First of all, tubular reabsorption occurs in the proximal convoluted tubule primarily. Okay? Actually, it technically occurs along the entire renal tubule, but most of it occurs in the proximal convoluted tubule. So for our argument, we'll just say that it occurs there. And the purpose of this is to take things that accidentally got filtered out of the blood. Okay, when we did the filtration process, we had to be indiscriminate. We couldn't say, oh, no, we'll keep hold of that glucose molecule. The glucose molecule followed where the water went. So did the sodium and potassium ions. And now we want to reclaim those glucose molecules of sodium and potassium ions and keep them in the body and not lose them in the urine. So this is the process by which I pull things back in, and water being one of the main things. And about 65% of everything that got filtered out at this point is going to get reabsorbed. There's a couple different pathways by which I can reabsorb things. There's something called a transcellular route and something called a paracellular route. So imagine that here I have my proximal convoluted tubule over here. This is where all the filtrate I just made occurs. And I want to pull substances out of the filtrate that I want to hold onto, and eventually I'm going to use the cells that make up the proximal convoluted tubule to do that, and then eventually I'm going to reintroduce my fluid back into something called the paratubular capillaries. Now, I have to back up here for 1 second, back to this picture here, and I need to tell you something, which is that if we look at our basic nephron here, one thing I forgot to mention before is that there's a network or net of capillaries that surround places like the proximal convoluted tubule, the distal convoluted tubule, and the nephron loop, and these are all called the peritubular capillaries, meaning that they're located peripheral to or next to the renal tubule. Okay, and that's this right here, these paratubular capillaries. So I'm going to take fluid that's in the glomerular filtrate and eventually introduce it back into the paratubular capillaries, return it to the blood, okay, from whence it came. Now to do that again, I told you there's two routes. There's a transcellular route, which means I literally go across the cell itself. So for example, I might use sodium glucose pumps to pump glucose actively into the cell and then back into the blood. I could use aquaporins, which are literally protein channels for movement of water, and allow them to pass through my cell and back into the paratubular capillaries. Or the other alternative is through this paracellular route, which means I literally go between two cells in the little space between cells, and a lot of water gets dragged in this way, and as the water gets dragged through between two cells, it brings many of its solutes with it. That's called solvent drag. Okay, when the water drags with it solutes like sodium ions or chloride ions, and in this case it can even drag with it urea and uric acid, which we really don't want it to drag with, but it just happens as part of the process. Okay. Now if we look at sodium, for example, we remember those sodium-potassium pumps that we learned about with your neurons and muscle tissue. Well, we have them also in places like your kidney cells, so that we can actively push sodium back into the paratubular capillaries. And when I when I pull sodium out of the cell and into the blood, that means that I'm left with a low concentration of sodium inside the cell, which means the higher concentration of sodium out here gets pulled down its concentration gradient into the cell and then pumped into the blood. Okay. Glucose is another one that gets pulled into the cell. It hitchhikes a ride with sodium through sodium-glucose transport proteins, which I believe I talked about in Bio 201. So, all in all, what do we actually reabsorb when all is said and done? Well, amino acids that we accidentally filtered out get pulled back in. They also hitchhike rides with sodium. Water, lots of water gets pulled back in, okay? And not only because of solvent drag, but also because, remember, those aquaporins, those water channels that are in the cells themselves. And by the way, remember you guys learned about antidiuretic hormone in Unit 1 when we were on the endocrine system? Antidiuretic hormone, if you remember, is that hormone that tells the kidneys to hold on to water. The way it does it is it tells the kidneys to add more aquaporins into the collecting duct here. The more aquaporins I have in this area, the more water I conserve. Okay. Now, next up, chloride ions. normally follow all the negatively charged stuff like amino acids and, all the positively charged stuff like potassium ions, calcium ions, sodium ions, hydrogen ions that are getting pulled back into the blood. So chloride follows because it's attracted to the positive charge. And many other electrolytes get dragged with the water back into the blood. Also, again, like I said, we accidentally, because we can't control it, do reabsorb anywhere from 40 to 60% of the urea that we just got done filtering out. So here it comes right back into the blood again. Though we didn't mean for it to happen, it did. And we also reabsorb much of the uric acid that we just filtered out. So the question is, well, geez, it seems like we reabsorbed almost everything. What didn't get reabsorbed? And the answer is, the big thing that didn't get reabsorbed is creatinine and at least some of the urea. Okay, so at least we have some urea that's in the forming urine now, and all the creatinine is in the forming urine. Now remember that this process of transporting things back into the blood has an upper limit, that the proteins that are being used to actively pump things such as the sodium-potassium pump to pump sodium into the blood can only do that so many times per minute. That's called the transport maximum. And an interesting permutation of this is an untreated diabetic. Imagine you had somebody with, say, type 1 diabetes and they were untreated, which means they've got huge quantities of glucose in their blood. That glucose is getting filtered out in the urine, and a lot of it's going to get reclaimed, as it should, but because there's so much glucose in the blood, that means there's far more glucose in the urine than there should be. And since my sodium glucose transport proteins can only transport so many glucose molecules per minute back into the blood, they literally get overwhelmed and cannot grab all the glucose that's passing by them and get it back into the blood. So glucose ends up building up in the urine, and of course, as you remember from our study of diabetes, one of the symptoms of untreated diabetes is glycosuria, or glucose present in the urine. Now, the next part of urine formation, we've already talked about filtration and reabsorption. The next part we're going to talk about is what happens in the nephron loop. And I remember I said there's two parts of the nephron loop. There's a thin segment called the descending segment, and it's permeable to water, which means water can get removed from the urine here, but it's not permeable to electrolytes, so things like sodium and potassium cannot exit. The fluid passes down through here. A lot of the water gets reclaimed. Then it goes up through the ascending segment, which is the thick segment. And here, this is impermeable to water, but it does allow for different types of electrolytes like chloride ions and potassium ions to be reabsorbed. So this is more reclamation. And we're going to learn that there's a special kind of nephron loop found in juxtamedullary nephrons that helps with some water conservation issues later on based on this same principle. Now in the last part here, in the distal convoluted tubule, we can have secretion occur. We can also have secretion occur in the proximal convoluted tubule and to a lesser degree in the nephron loop. During secretion, we have one more chance to actively grab things out of the peritubular capillaries. Remember, the peritubular capillaries surround all of this here. There's peritubular capillaries surrounding the convoluted tubules here. This is our one last chance to actively pull stuff out of the peritubular capillaries and put it into the forming urine. We can literally secrete substances from the blood and secrete them into the forming urine. This is where we get one more chance to grab urea and uric acid, ammonia molecules, drug molecules like penicillin and aspirin, all of those different things, and hydrogen ions if we need to adjust the blood pH. Those are all things that I can actively secrete into the forming urine. I pull it from the peritubular capillaries and I actively secrete it into the urine. Now, in the distal convoluted tubule and the collecting duct, I can also do this, but in addition, I can also actively, again, still keep reabsorbing water. And this water reabsorption can be controlled hormonally. Remember, aldosterone induces the kidneys to absorb more sodium, and where sodium goes, water follows. That antidiuretic hormone causes cells in the collecting duct to install more aquaporins in the cells, which means there's more holes available for water to be pulled out of the forming urine. And by the way, if you've ever woken up after drinking all night, drinking alcohol, you wake up with a headache, that's because you're dehydrated. And the way that alcohol dehydrates you is that alcohol inhibits antidiuretic hormone activity. So it inhibits your body's ability to hold on to water, and instead the fluid exits out of the body as urine, and you end up dehydrated and with a headache. Parathyroid hormone also influences the kidneys and encourages them to hold on to calcium and to release phosphorus. So that's another thing that can be actively held onto while secreting the phosphorus. All right, the last part of urine formation is how we conserve water, and this is really critical because remember again, your body is filtering hundreds of liters of blood plasma per day and about 180, and yet it is only producing about 2 liters of urine, which means nearly all of the water volume that is being filtered gets returned back to the blood. And we need to figure out exactly how we go about conserving this water because we cannot conserve all of it just by reabsorption in the proximal convoluted tubule. And this is where we really involve the juxtamedullary nephrons. Remember, those were the rarer of the 2 types of nephrons. They're found right at the juncture between the cortex and the renal medulla. And they have extremely long nephron loops that are, in fact, as long as the renal pyramids are, which is very, very long. And these very long nephron loops do something really critical. What they do is they create an environment that has a high salinity. In other words, it's very salty. And that very salty environment is important because if you remember, if I have all my cortical nephrons up here in the cortex, okay, they all feed into a common collecting duct, and that collecting duct then travels all the way down to the bottom of the renal pyramid where it dumps its forming urine into a minor calyx through the renal papilla. And as this collecting duct travels through the renal pyramid, it is the juxtamedullary nephrons that have really long nephron loops that dip down like this that are creating this highly saline environment, which I'll represent with green here. So that as a collecting duct over here has its forming urine passed down through this very, very salty environment, what happens is the water in the urine inside the collecting duct leaves the collecting duct to try to dilute out the highly saline environment that surrounds it. And that's how we basically conserve the majority of the water. Now I want to take a second and just look at exactly how it is that we make this very saline environment. And imagine I'm looking at one of the nephron loops from one of these juxtamedullary nephrons. This is one of those long nephron loops that dips way down into the renal pyramid. On the way down, let's say I have a solute concentration value of 300. Notice that as the urine passes down through this nephron loop, that the concentration of the urine gets steadily more concentrated. What that means is we're losing water, okay? And that's because this thin segment here, as we said before, the thin segment is permeable to water, and water is going to exit out because it's salty around here and is going to try to dilute out the saltiness. Then as we come back up the thick segment on the other side, this is not permeable to water, but it is permeable to all these electrolytes like sodium and potassium and chloride ions, and they begin to exit to try to dilute out the water. The end result is by the time the urine leaves this water conservation area, it's not as concentrated in solutes, which means the solutes are out in the area here, making it very saline, so that when the collecting duct here passes its urine down through this very saline environment, the water in the collecting duct exits out to try to dilute this saline environment. Now that's a pretty complicated process. Here's what I want you to get out of it. Remember the difference between a cortical and a juxtamedullary nephron, and that cortical nephrons are all about making urine, and juxtamedullary nephrons are really about making this very saline environment, so that when collecting ducts pass down through the renal pyramid, water is conserved or pulled out of the forming urine in that collecting duct due to the very saline or salty environment. Okay, now this whole process of how this juxtamedullary nephron creates this very salt environment is called the countercurrent multiplier.